And the first things which came to me was that this guy is interested in life. And indeed, his career is blossoming, moving on with career. And what I can say about Hendrik is that he's extremely creative person. And that's not very often, but so essential in science. So Hendrik, I know you're doing marvelous things in Basel, and we wish you all the best. And thank you for joining us today. And now I will go to Cesare.
Hello, everybody. Can everyone hear me well? My name is Cesare Ritz, and I am a postdoctoral researcher at Dorota Skowrońska-Krawczyk Lab at Gavin Herbert Eye Institute here at the University of California, Irvine. It is my great pleasure to welcome you to today's talk and to present you with our distinguished speaker, Professor Hendrik Scholl. Professor Scholl is the director and the co-founder of the Institute of Molecular Ophthalmology and Clinical Ophthalmology in Basel, in Switzerland. He serves also as a professor and chairman of the Department of Ophthalmology at the University of Basel in Switzerland. Dr. Scholl received his medical degree from the University of Tübingen in Germany before completing his ophthalmology residency at the same institution and his clinical research fellowship in medical retina at the renowned Moorfields Eye Hospital in London, in the United Kingdom. Professor Scholl then joined the medical faculty at the University of Bonn in Germany, where he served numerous faculty positions and served as a consultant ophthalmologist, vitreoretinal surgeon, and research coordinator. In 2010, Professor Scholl moved to the United States, where from 2010 he served as a professor in ophthalmology, and from 2011 as an endowed chair at Wilmer Eye Institute in Baltimore, Maryland. During his time there, Professor Scholl headed the Retinal Degeneration Clinic and the Visual Neurophysiology Service of the Johns Hopkins Hospital, and was later appointed as a co-director of the University's Center for Stem Cell and Ocular Regenerative Medicine. Throughout his career, Dr. Scholl received numerous awards, including European Vision Award in 2008, the American Academy of Ophthalmology Achievement Award in 2019, and the Paul Henkin Award of the Macula Society in 2020. In his clinical work, Professor Scholl specializes in treatment of retinal diseases. His research goals at the Institute of Molecular and Clinical Ophthalmology in Basel are focused on retinal neurodegeneration with the goal of developing novel therapies, including molecular cell biology, gene therapy, and stem cells. Professor Scholl has co-authored or authored more than 200 peer-reviewed papers. Without further ado, please welcome Professor Hendrik Scholl.
Yeah, thank you very much for the kind introduction. I hope that people can hear me and can now also see the first slide. I'm very happy and honored for the invitation to speak today. It would have been even nicer to be in Irvine, California right now. But I can say from the distance that I have been a fan of Chris Palczewski's work over the last decades, really. And I'm proud to be one of the few who can spell his last name. I have a few tricks, certainly, how to accomplish that. So it's a pleasure to present on the topic disease stage specific therapies for inherited retinal degenerations. Just briefly, my financial disclosures. It was mentioned I'm the co-founder and scientific co-director of the Institute of Molecular and Clinical Ophthalmology Basel, or briefly IOB, which is constituted as a non-profit foundation and receives funding from the University of Basel, University Hospital Basel, Novartis, and the government of Basel-Stadt. When we look at the problem, actually, what are causes of blindness and what's the role of inherited retinal dystrophies? we see that when we look at the age group of 20 to 60 here, the cause of blindness in Germany, representing really the Western industrialized countries, then the group of inherited retinal dystrophies are the largest single group. About a quarter of all cases of blindness go back to inherited retinal dystrophies. The other portion on the upper left, these 37% is a mixed bag of all sorts of causes of blindness. That picture changes when we go to the very old. In that moment, 2/3 or so are due to age-related macular degeneration. Speaking about inherited retinal degenerations means that we have to look at the causative genes, and there are many. There are actually now 280 identified genes that, if mutated, give rise to inherited retinal diseases. That leaves us as clinicians with an enormous complexity of these genes and the respective phenotypes. And we also need to know that different mutations in those genes can be correlated with different phenotypes also. So it's quite a significant complexity that we have to deal with. Very recent data now on just autosomal recessively inherited retinal diseases show that almost 6 million people are expected to be affected. And the genetic prevalence of autosomal recessively inherited dystrophies alone is approximately 1 case in about 1,500 individuals. What is stunning, and I'm still digesting that, is that 36% of the world population are carriers of at least one mutation that would cause autosomal recessive inherited retinal dystrophies if they had inherited 2 mutations. So I think this is something quite important to keep in mind, that almost 3 billion people worldwide carry at least one mutation of retinal dystrophies. On that slide, we see the therapeutic window of opportunity. We have to. I mean, that is worth a comment here. There's very little therapy for those conditions as we speak. So there are many developments, and I group them here in that figure, and their role depends on where we stand in the progression of the disease that we see on the x-axis, the progression of photoreceptor cell degeneration, which means when we go to the left, there's still significant photoreceptor viable cells available. When we go to the right, then we lose them over time, and eventually there's very little left. So when we look at situations where there is still significant photoreceptor cells available, we can consider therapy approaches such as gene replacement therapy, pharmacotherapy, neuroprotection. And then if we get to end-stage disease, then there are approaches such as stem cell therapy, retinal prosthesis, but also optogenetic therapy. And today I would like to discuss 2 examples, one more to the left, one more to the right. I would like to speak about pharmacotherapy in the beginning, and at the end about optogenetic therapy. But then the question is, what is the disease we actually should discuss? And again, I refer to this paper in PNAS by Carlo Rivolta and colleagues from IUB about the genetic prevalence of autosomal recessively inherited retinal diseases. And by far the most prevalent disease gene is ABCA4, mutations in which are responsible for about 1/3 of all IRD cases that are recessively inherited. It's unfortunately a complex gene, has the highest number of mutations in populations of European descent, and a cumulative carrier frequency of 2.5%. The expected genetic prevalence of Stargardt disease, which is the disease that we will talk about in a moment, that is due to mutations in that disease gene, ABCA4, is estimated to be 1 in 6,500 cases. This is pretty new data, which is significantly more frequent than we thought. The typical number that is being discussed is 1 in 10,000. But we know more and more that this disease is not that rare as we thought before. So what is Stargardt disease? For the non-clinicians in the room or in the meeting, is the most common juvenile macular dystrophy. It's about 7% of all inherited retinal dystrophies. It shows autosomal recessive inheritance. Patients report reduced central visual acuity, blurriness, blind spots, and delayed dark adaptation. And this is due to loss of central vision due to retinal atrophy. The manifestation age ranges from pretty early, such as 5 years, to patients that are older than 40. I have at least 2 patients that are asymptomatic at age 70, where we diagnosed Stargardt disease. So it's a wide range of manifestation age. The ocular exam signs that you see an example here of a right eye of a Stargardt patient here to the left are areas of retinal atrophy in the center. And then these subretinal yellowish flecks that are more peripherally. And they are quite important because they indicate accumulation of bisretinoids at the level of the retinal pigment epithelium. And they indicate an important element of the pathophysiology. I will get to that later. There's a whole spectrum of the phenotypic appearance of ABCA4 associated retinal dystrophy. So I mentioned ABCA4 associated retinal dystrophy because there is more than typical Stargardt disease. So typical Stargardt disease would be in panel D. It's controversial if there is an overlap with age-related macular degeneration. I rather do not believe that. I believe these are patients that are misdiagnosed. They look at, for people that are not experienced, these retinas may look like AMD, but are indeed Stargardt disease. But there are other phenotypes, such as cone rod dystrophy, that we see that is due to ABCA4, and also very advanced disease that could be indistinguishable to retinitis pigmentosa. I do not believe that ABCA4 causes retinitis pigmentosa. Retinitis pigmentosa being a disease that I discuss later in my presentation, that is really a rod-driven disease, but the end stage can look like in retinitis pigmentosa. So what happens over time is that we see atrophy of outer retinal layers. This is from a review article by Franz Kremers. And we see in the first column relatively early disease, but already when we look at OCT, we see some atrophy right in the fovea. Then we get to the second column where we see chorioretinal atrophy. The third column already shows advanced chorioretinal atrophy. And then in the fourth column, we see this end-stage disease where we even look at sclera because there's nothing left in between, such as outer retina and retinal pigment epithelium. The question is, what is the natural history? The question is, how does the disease develop? And if we intervene at one stage, can we make an impact on the disease progression? And this has been really unknown and prompted Foundation Fighting Blindness to really launch the first observational study that would be conducted like a clinical trial, like an interventional trial. And at the time when I was at Hopkins, we discussed with Foundation Fighting Blindness, we found funding for the study. And then we designed the study and implemented the study. And the study is called ProgStar, the Natural History of the Progression of Atrophy Secondary to Stargardt Disease Study. And in ProgStar, 7 centers in the United States and 3 centers in Europe, if you consider the UK being part of Europe, of course, have collaborated to contribute cases to be followed longitudinally over time. And we had a central reading center at the Doheny Eye Institute and a central coordinating center at the Wilmer Eye Institute. The study chair in 2016 actually moved from Baltimore to Basel, Switzerland. So in ProgStar, we had a retrospective arm where we included about 350 patients or so, and a prospective arm. In the prospective arm, we recruited 259 patients. You see the centers that contributed here and the number of patients that went into the longitudinal prospective longitudinal study. We enrolled patients that carried at least 2 pathogenic mutations in the ABCA4 gene. They needed to be at least 6 years old, needed to have at least 1 well-demarcated area of atrophy used on fundus autofluorescence, needed to have vision equal or better than 2400. And of course, at the time, no inclusion in clinical trials, but at the time, there were no clinical trials when we actually launched the ProgStar study. I show that multi-panel figure again to make the point that in ProgStar, we included these patients, which means not very early, but also not very late. We included those patients because they had features, and I will get to that, using autofluorescence imaging that could be tracked over time and could be read by a reading center, could be evaluated, could be demarcated by a reading center, like in a clinical trial. So this is the design of the prospective ProgStar study. We had different technologies that we have been using that are shown here, like microperimetry to characterize how well a patient would be able to see. And then OCT, that I will not discuss today. We segmented all the retinal layers in every second B scan of all the 49 B scans that were recorded per eye, per patient, per visit. So it's quite a large data set. Autofluorescence images, and we also recorded electroretinograms to characterize the baseline if patients either have a pure macular phenotype or have more widespread disease, such as cone rod dystrophy. And we also recorded, of course, visual acuity. And I would like to briefly comment on visual acuity. So this is the visual acuity data in the prospective cohort. We had 434 study eyes that we could evaluate. And we grouped them according to WHO criteria, so the level of vision that they had when they entered the study, and how visual acuity changed over time. And this is what we found. We found that the overall rate reached statistical significance, although it was just half a letter on a visual acuity chart. And we used the ETDRS acuity chart, and all the clinicians would know it has 100 letters. So, and just half a letter changed over time, but it reached statistical significance, which just speaks in favor of the methodology that we used in a multi-center, in this multi-center design. So we rigorously measured visual acuity, but I think there's agreement that half a letter of visual acuity is clinically not significant. This is almost nothing, but at least this is what we found. But what is interesting is that when we grouped them before and followed them over time, that patients that started with pretty good visual acuity, they lost visual acuity at a rate of about 1.5 letters per year. Then patients that started with mediocre visual acuity, they declined at a rate of about 2 letters per year. That was highly statistically significant. That also applied to the patient group that was close to blindness, according to the US definition, although they only lost half a letter or so. And what was interesting was that those patients that started worse than 2200 showed an improvement. And it was not just a chance finding, this also reached statistical significance. So it was not just a random effect. So there was some improvement for those patients that started so with such a bad visual acuity. There are many reasons that could explain that effect. It has to do with fixation stability, the number of so-called PRLs, preferred retinal loci, outside the foveal center, and so forth. But it's an important effect, and such an effect should be kept in mind when we look at clinical trial data. For example, the stem cell trials that were published on Stargardt and AMD, where you see such an effect that patients gain a line or so when they start at 2200 or worse. And keep in mind, this is just a natural history. So I think this should be kept in mind evaluating future clinical trial results, that would be an actually expected and not unexpected situation, that patients may gain not much, but half a letter or so per year in the wrong direction, if you will, because they improve a bit. An important finding of the study was to estimate how long does it take to lose 3 lines on a visual acuity chart, because this 3-line criterion is known, because that's a criterion by FDA that would define a significant visual acuity gain or loss. 3 lines on a visual acuity chart. And our estimate from the ProgStar study, it's 27 years. And that means we simply cannot use visual acuity as a primary outcome measure, because that is an unbearable duration for any clinical intervention to show either improvement, but here more importantly, a lack of or a slowing down of degeneration over time. So that was one of the conclusions from ProgStar, that the visual acuity cannot be it, and we provided the data to support that. The most important and the primary outcome measure was fundus autofluorescence imaging. So we looked at fundus autofluorescence imaging. We measured these lesions that we also defined in the ProgStar study. So this was published as ProgStar report number 11. We reported data of 488 eyes that were included in the data analysis, and the final data set comprised of 432 eyes. We define these lesions. Here we look at this marked in with a blue margin here, these lesions that we called definitely decreased autofluorescence, DDAF. This is a level of darkness close to 100% in reference to the optic nerve head, that is with a white arrow. And then there were other lesions that we coined the term questionably decreased fundus autofluorescence, where we found between 50 and 90% darkness. So we looked at DDAF and QDAF over time. So what we found was that the change over time in the area of DDAF was about 3/4 of a square millimeter per year. We also found that this was highly dependent on baseline area, relatively similar, but somewhat different than what we find in the field of late dry AMD. So you see here, when we start at no lesion or very little lesion, we see a progression rate of less than half a square millimeter per year. When we have these medium lesions, then we are close to the overall mean, namely about 3/4 of a square millimeter per year. And then larger lesions grew even faster, almost 1.5 square millimeters per year. The measurements appear to also depend on unifocal versus multifocal lesions. We see on the left estimated progression for purely unifocal lesions that was not as fast as lesions that were multifocal in nature that we see on the right. Overall, the progression rate is somewhat consistent to other monocenter prospective studies that we have seen in the field, but dissimilar. It's really dissimilar, so different to what we see in late atrophic AMD. In late atrophic AMD, we see more than double the speed of progression as compared to Stargardt disease. So Stargardt disease in this regard is different to late atrophic AMD. So back to that slide. So now that we defined outcome measures for Stargardt disease, we defined how this can be measured. We defined how a clinical trial could be designed in order to measure therapeutic effect. And this was actually approved by FDA and is now being used in almost all clinical trials that are currently taking place for Stargardt disease. And I would just like to show one of them. Keep in mind, again, this multi-panel figure, that all clinical trials like the PROXSTAR study look at this specific slice, so to speak, of the natural history. Not very early cases, but also not very late cases, where you see these areas of chorioretinal atrophy that can be tracked over time. When we get to therapy development in Stargardt disease, specifically compound development, pharmaceutical compounds for Stargardt disease, we have to look at this at the visual cycle. It's a bit embarrassing to speak about the visual cycle in a meeting that is organized by Chris Palczewski, because he is the master of the visual cycle and characterized many important molecules here, such as RP65, even cured ABCA, ABCR, or ABCA4 knockout mice, both with gene therapy and with pharmacotherapy, which is also would be an interesting separate topic that I will not cover today. But of course, RP65 plays a role. There's even one company that would block RP65 in order to slow down the visual cycle to have a therapeutic effect in Stargardt disease. Then there are others that block RBP4 to slow down the visual cycle. There are aldehyde traps to lower ultrans retinal. That's another approach that was developed by Chris Palczewski in his lab. And another approach that is deuteration of vitamin A in order to simply by chemical reactions slow down the formation of bisretinoids at the level of the retinal pigment epithelium. So I spent a couple of slides on that one, although I don't have the final slide deck because it's always complicated to deal with companies in that regard. So I'm the coordinating PI on this clinical phase 2 trial that is completed. The final data set was once presented at the Retina Society, but I will not present the final slide deck here. But I would like to explain what is this deuterated vitamin A about. So it's selectively deuterated at position 20 of the vitamin A molecule and can as such be taken orally once a day. It slows the vitamin A dimerization by 4 to 5-fold, and then in vivo leads to a reduction of vitamin A dimers of about 80% after 3 months of treatment in an ABCR or ABCA4 knockout mouse model. This was then studied in further detail by Peter Scharbel-Issa at Oxford University, published in PNAS a couple of years ago. Here you see the relative fundus autofluorescence intensity level on the y-axis. So fundus autofluorescence as an indirect measure of bisretinoid signals that are measured by this imaging technique. So example of these images are shown on the lower right. And the relative fundus autofluorescence intensity was measured over time. So what you see the two extremes. One extreme is this dotted line. These are wild type mice on a regular vitamin A diet. and then the line that is purely green are ABCA4 knockout mice on a regular vitamin A diet. And now you have all combinations. You have the that are shown in orange, mice that would receive the compound. It's called ALK001, so deuterated vitamin A. And you see that if mice would get the compound from the beginning throughout the study period, they would be almost indistinguishable from wild-type mice. When they get the compound later, you still see a significant treatment effect. You even see the treatment effect when you go in as late as 6 months and only observe for 3 months. Even then you see the treatment effect. And I think this shows how nicely reproducible that compound would be on the relative fundus autofluorescence intensity level, which means on the bisretinoid level at the level of the RPE. That prompted a clinical trial called the TEASE trial, which is a phase 2 placebo-controlled, double-masked clinical trial to look at efficacy and safety of this compound in 20 normal subjects and 30 subjects with in 20 sorry, I have to say this is all in Stargardt patients, 20 Stargardt patients on placebo and 30 patients on the accused compound. At month 12, 10 out of the 20 Stargardt patients were randomly assigned to the compound after 1 year. And the primary efficacy endpoint was the growth rate of atrophic lesions that was pre-specified according to the ProgStar study that we coined DDAF. And here you see that. So these atrophic lesions were measured. They expand over time in Stargardt disease and were accepted by the FDA as the primary endpoint of that clinical trial. Without having the final data set, I can tell you the clinical trial reached its primary endpoint, and there will be more about this compound in the clinical trial in the near future. So that was about Stargardt disease. In the second half of my presentation, I would like to speak about Retinitis Pigmentosa. This may be known to many of you in the auditorium and in the meeting. It's a term given to a set of inherited retinal diseases that feature degeneration of rod and cone photoreceptors with a worldwide prevalence of about 1 in 3,500 for a total of more than 2 million affected individuals throughout the world. We see a degeneration of the outer retina in the disease. Here, this is showing schematically in the middle. Patients retain ganglion cells, bipolar cells, horizontal cells, bipolar cells, and many of them retain cone cell bodies in the outer retina. So this is how a normal retina would look like for a clinician. You see the optic nerve, you see the retinal vessels, you see the center of the retina with the macula. And I'm actually quite happy about this retina looking normal because it's actually my retina. And this would be a patient with retinitis pigmentosa, and you see why the disease is called retina pigmentosa, because you have this pigment migration from the retinal pigment epithelium into the retina that then leads to these black bone spicule-like pigmentations that you see typically in the mid-periphery of these patients. These patients are night blind, and then they start losing the mid-peripheral visual field and eventually develop tunnel vision. And I would like to simulate that here. So these patients, as opposed to Stargardt disease, where you have a macular phenotype, where you have very good mobility, very good orientation, but you can't read and you cannot recognize faces. These patients, they have to scan. They have to scan around. They have very little contrast sensitivity, but acuity may not be that bad. But the problem here is that eventually they may lose vision completely. And many patients do. And the question then is, what can we do about that? And one answer would be optogenetic therapy. So optogenic therapy, there are different approaches that can be used. These were reviewed by here Jose Sahel and Botondroska a couple of years ago. You see, excuse me, the intact retina on the left. Then you see on the right, you see the GenSight approach, targeting ganglion cells in the inner retina using an intravitreal injection. And in the middle, you see the IOB approach, where we target the remaining cone cell bodies that we see in many patients that develop bare light perception or no light perception secondary to retinitis pigmentosa. So optogenic therapy has already reached the clinic. You may have seen this paper that was published, I believe, in August or so in Nature Medicine, from the study of the GenSight compound, the GS30, published in Nature Medicine, where the first patient that was treated with the compound after significant rehabilitation was shown to have restoration of some vision. And that was very important for us and for the field, because it showed that optogenetics can work, even if you just, if I may say that, just target the ganglion cells. Because the big problem with ganglion cells is they are arranged as a ring around the fovea, while with the approach of targeting remaining cone cell bodies in the retina, that would allow to target cells that are actually made to see. In this case, they have obviously have lost light sensitivity because they have lost outer segments, but they are arranged as a grid in the fovea and can be targeted. We call them dormant cones, and we target these dormant cones because that should allow to restore features of retinal computation, because the inner retinal circuitry can then be reactivated. This all goes back to the proof of principle that was shown by Botondroska in a mouse model 10 years ago or so, where they targeted these dormant cones in the outer retina and could restore light sensitivity in these mice. At IOB, we have access to specific technology platforms that we use to develop that compound. One is our cell-specific targeting by promoters that we published 3 years ago in Nature Neuroscience, where we can essentially target any cell type in the retina, and not only in mice. We have shown that in non-human primates, and most importantly in humans. As an example, when we use the ProA7 promoter, we can specifically target cones. In the middle, you see the optogenic tools, and on the right, the AAV capsids that we use for experimentation. And the clinical candidate optogenic vector can then be looked at in its expression in several different model systems that we use at IOB. Of course, mouse retina in the first row, then retinal organoids in the second row, ex vivo human retina that we obtain from multi-organ donations, and in vivo and in non-human primates. Since I'm the clinician here, I speak about our clinical multi-center study that we are running in parallel. We developed a method for pre-screening of these patients that are potentially eligible for this cone-based optogenetic vision restoration. And we are currently leading a multi-center worldwide retrospective imaging study that we call the ICONIC study, to determine the cone volumes in patients with very low vision. And you see the centers that are currently participating in this effort. We include patients that have very low vision, that have no detectable ELG, that are genetically confirmed, although our therapy would not be gene specific, but currently we would like to learn there may be specific genotypes that may be more or less suited for this approach. These patients need to have healthy inner retinal structure and optic nerve, because of course they would need to have tissue available that would allow that the visual signal would travel along the visual pathway to the visual cortex. Exclusion criteria include media opacity, of course, that would prevent OCT imaging, or significant nystagmus that would prevent to obtain at least relatively good OCT scans, other eye diseases that would affect optic nerve, or, for example, cystoid macular edema. So what are we doing? You see on the left a patient with complete foveal atrophy that would be not a good candidate for our approach. And then in the middle, you see two examples of patients with Leber's congenital amaurosis. And here you see that the fovea retains significant structure, and those patients should be good candidates for our approach. In our workflow, we train an artificial intelligence algorithm to segment the total retina and OCT scans, and then we reconstruct the total retinal volume, and we determine the minimal thickness point in the foveal center, and then take the surrounding 350 and 700 micrometer hypercylinder, and we use this measure of volume as a proxy of the overall cone volume that remains in the fovea. We first did it by segmentation using the Heidelberg machine. The problem is if there is significant degeneration in the retina, these segmentation typically fail in low vision patients. So we developed our own proprietary software tool that now allows us to segment these retinas quite reliably. And here you see these the results. These are these volumetric total retinal reconstructions that are based on this AI segmented retinas. On the left, a normal retina. In the middle, a patient with foveal atrophy that would not be eligible. You see this let's call it steep valley in the very middle, and then a patient with low vision that has a preserved fovea that would be eligible. So we calculate the foveal volumes based on the minimal thickness point, and we have an approximation for the remaining cone mass. And then we look at structure-function correlation. And here, what you see on the x-axis is foveal volume, and on the y-axis you see visual acuity. It's logMAR, so which means the higher the logMAR, the lower the visual acuity. And then you see this group of green dots. These are normal subjects. Normal subject meaning they have normal visual acuity and they have very good foveal volume. This is normal. And then you have patients where you have low acuity, but you also have degeneration, so they have low foveal volume. But then you have this group that we here circled here in blue, and you see that these are low vision patients that have significant foveal volume left. And this is our target population for our approach. So some patients show this, we call it structure-function discorrelation, which means they have low vision despite preserved fovea. And these patients are excellent candidates for our approach. And our estimate currently is that it's about 20 patients of all patients with retinitis pigmentosa or inherited retinal dystrophies that have very low visual acuity, have such foveal sparing and would be candidates. And here are shown two examples. You see a healthy subject in the middle, and just above you see an example of such a patient. No light perception, it's quite amazing, but nicely preserved foveal volume. So in our ICONIC study, we currently are looking at these retrospective images. and we plan a prospective study and look at the natural history of these patients, and also start recruiting them, because the next step will be our first-in-man phase 1b/2a clinical trial, evaluating the safety and tolerability of the cone-based optogenetic vision restoration approach. And we anticipate that to happen in about 18 months from now. And of course, we aim to do that in Basel at IOB, and we're currently preparing for that clinical trial. So that brings me to the end of my presentation. I think I'm relatively well in time, but I would still like to thank Chris for the invitation. Here you see the two of us in Basel at the International Conference on Molecular Systems Engineering. The visit has come to a temporary end because of the pandemic, so we have to resuscitate that, and we are already discussing how we should do that. I'm very much looking forward to it. I thank everybody for the attention. I'm very happy to take questions.
This was truly wonderful, and nice to see this application of what we have discovered about the diseases and understanding the pathology. And now you're taking this next necessary step. So guys, if you have any question, please drop me a note in Dropbox. Send it to everybody. I have a different computer, so send not just to me, but to everybody. Corinne.
Yes, wonderful, wonderful presentation. I was wondering about the vectors that you made. Are they available for general public consumption, or how does one get to the, specifically targeted AAVs that you have built?
Yeah. So the final compound I've not shown. So the patent was submitted. There are various components. Some of the components would simply be available. Some are not. So I can just not disclose it here publicly, but we can start chatting about that in a one-to-one discussion, and I can tell you what would be available and what not.
Thank you.
Hey, John.
Oh, hello, Dr. Scholl. Amazing talk. I learned a lot. I was wondering about Stargardt's disease and its progression to the many different disease phenotypes. I was wondering if you could elaborate a bit more, and if you see any definitive patterns, and if it's like what sort of environmental factors play a role in one phenotype versus another taking place. And my second question is for the cone-based photoreceptor optogenetics. Do patients, are they able to visualize the world in color, or is it still in sort of like a black and white?
Yep. Let's get to the first question first. I mean, the question that you ask may have to do with the representativeness that we have. If we see a therapeutic effect, for example, when we use a compound like deuterated vitamin A, and that is a question that you always have when you run clinical trials. The clinical trials are quite an artificial endeavor, and because you have very strict in and exclusion criteria, and still you would like to develop something that then would help the total population out there. The question is, when we look at small lesions of choroidal retinal atrophy and we find a therapeutic effect, would that therapeutic effect still hold if we get to more advanced disease or to very early disease? The thing is, we do not know. This is what then happens after clinical trials and market authorization. This is what happens actually in clinic. And this is actually the role of clinical sciences, to make observations and of course use these therapies for patients that are far beyond what actually happened in the clinical trial. And this is a typical pattern that we see. Clinical trial, relatively narrow patient population, and then later in therapeutic reality, you have a larger variability of affected patients, in our case, phenotypes. And then the question is, how applicable is the therapy for these phenotypes? My guess is that when we get to very late disease, all these chemical compounds may have very little effect. When we get to very early disease, those compounds may have an even better effect than in these patients that are currently being studied in these clinical trials. The second question is, if in our or other optogenetic therapy trials, patients would then actually see color. I have to say that's very demanding. Keep in mind that color perception depends on disproportional photon catch relative to the photopigment that is being used. For our approach to cone optogenetics, but also other approaches, there's one photoswitch protein that has one specific peak of sensitivity, and which means it's absolutely impossible that these patients would have a colorful representation of the outer world if the therapy works. I have to say, this is now having my hat of a clinician on. When we speak with patients that have achromatopsia, yeah, we think or we discuss achromatopsia as if the biggest problem of these patients is they do not see color. It's absolutely not the case. The problem of these patients with their achromatopsia is that they have a visual acuity of 20/100. They are not keen on seeing colors, I tell you, right? You can see a traffic sign perfectly fine if you know the order, right? If you have no color vision. But if you have 20/100 or lower visual acuity, you're very handicapped. So keep that in mind, that color vision is it's very demanding, yeah, to achieve that. I think that's not achievable, but I would also say it's not important, not that important.
Thank you. Thank you so much.
All right, let's move on to Professor Vice Chairman Vladimir.
Thank you, Dr. Scholl. This is really amazing work, beautiful and very, very exciting. I have a question for each part of your talk. For the Stargardt disease, you showed that the ALK compounds prevent autofluorescence. Do they also provide functional and structural protection to the mouse retina? And the question for the second part is with the optogenetics, do you think that rescuing cone function will also in the long run prevent the cones from dying, or is this just a way of extending the function of cones, but eventually they are still expected to degenerate?
Yeah, excellent question, right? I have to say, so we can discuss for hours now. These are very relevant questions and they are very important. So the first question is mouse models of Stargardt disease. I mean, again, Dr. Palczewski is the expert here. The question is, does the single ABCA4 knockout mouse actually has a phenotype? In my opinion, I mean, you speak to a poor clinician here, that's debatable. I think that was the reason why Chris started to work with a double knockout mouse in order to have a phenotype that you can then show to rescue if you intervene. Yeah. So in my personal opinion, I would say the standard knockout mouse model, ABCR or ABCA4 knockout mouse model, doesn't really have a phenotype. Yeah. So therefore, all these claims that a mouse model is being rescued with these compounds, I'm not sure how valid they are. But we are now in clinical trials. There are more than half a dozen compounds now in clinical trials, and I think we have to make observations there. To your second question, to date, it has not been shown for any gene therapy trial, including the RP65 gene therapy trials or Luxturna, that it has any effect on the survival of the target cells. Yeah. And if you ask me, I'm somewhat pessimistic that it actually has, right? Because if we had seen that Luxturna would have an effect, it would have been reported. I would at least guess so, right? So I'm not sure that you actually can modify the survival by repairing the genetic defect. Okay, here with optogenetic it's different. Here we transduce and have the expression of a new gene, of photoswitch gene. Does that have any effect on the survival of these cells? We do not know. We do not even aim for that, but it's at least conceivable that it could have an effect, because those cells at least are then closer to the physiology than before, because they fire and are connected to secondary cells. But it's a good point. It's still conceivable that we would intervene, and the intervention would not last for decades. It would last for very long, I would guess, but maybe not until the end of the life. That's true.
Thank you.
All right, let's move to Brother Anand.
Yeah, hi, Hendrik. That was beautiful. I'm so happy to see that all this basic research done by Chris and many others is now sort of leading to some sort of clarity and maybe therapy. But, you know, Vladimir stole part of my question, actually. I was sort of going to ask you that if these cells, so now I'm going to modify the question a little bit. If these cells, if these cones or any other cell type are not going to survive for too long, and they're going to survive for a couple of years, do you think, you know, spending millions of dollars per dose or whatever that is being done, say for RP65, is going to be worth it? Yes, of course, for a few years, it may give you a benefit. Why not we think of therapies that allow photoreceptor cells to survive longer, rather than repairing them or treating them in a temporary way? Why not look for something that can keep them alive? And that could be a much more generic general therapy for many different kinds of retinal degenerations, right? Rather than having gene therapy for each one of them individually and taking years and years of trials, and something will come out only for a few years. But, you know, I see around me literally so many researchers going after something which is not going to be permanent. I mean, that's what you just said too. And that's what is shown by Sam Jacobson and others, and Art is here and others as well, that these cells don't survive. They are still going to die.
Yeah, but so, I mean, that was the reason why I looked at the title of my talk, right? So yeah, I absolutely do not disagree that it's worth looking at the first part also, or even more at the first part, where there is not such widespread degeneration, right? And we may see greater therapeutic effect, at least on a population level, if we target those. And I agree, certainly. And you have seen that we are working on that also. But on the second part, it's debatable how long those cone cells survive in the retina. When you look at Dr. Mylam's papers, even in end-stage retinitis pigmentosa, she always found at least one layer of cone cell bodies in the outer retina, which would mean that therapy not only would last very long, but could even be applicable to all patients with retinitis pigmentosa that have lost vision. We did a very conservative approach where we have definite outer structure left in the retina, and we target these patients first. But when you look at histology, it could be all patients. And the question is, how long do these cone cells survive? And it could be indefinitely. I mean, not indefinitely, right? But until the end of the patient's life, because they appear to be very resistant. It's unclear, I'm not a cell biologist, but it's unclear why rod cells die so easily and so early. And why then cones are affected, that is not understood. And then if cones are affected, why they lose outer segments, and then why would they then still stay in the outer retina? But all of that is fact. That's all fact, right? They are secondarily affected, then they lose outer segments, and then they stay there. We do not know. I still believe, and that has to do with retinal computation or the computation of the visual signal. I believe, and we have seen that in the retinal prosthesis world, that if you target outer retina and if you target the center of the retina, then you take advantage of all the retinal computation that takes place in the remaining retina, and you have a cortical magnification factor that may have a very important effect on vision restorations in these patients. But it remains to be seen. We have to run a clinical trial.
So if I may, Hendrik, comment on what my brother says, because I agree with him 100%. And you know, we were trying to and still working on pursuing the systems pharmacology. So if you take a patient with RP at young age, in many cases, not in all, but many cases, they still have a functional retina. And it is the degenerative process. So what it makes for the young body, the young organism that survives this insult of mutation, but yet it cannot cope for a long time. So if we can identify this, we as a field, that there are signals from those cells, for example, excessive signaling pathways and neurotransmitter release, if we can dampen that in a prophylactic way and extend perfect vision for quite a while, even if this mutation is present. Can you comment on that? What is your thought? Is it not where we virtually should put all our effort into that?
So I can put my scientific hat on, and I can put my clinician's hat on. And I put now my clinician's hat on because on the scientific part I agree. But when I put my clinician's hat on, I think about how clinical trials are being designed, how are they approved, how are they being seen by regulatory agencies. And in the moment when you get into, let's call it prophylaxis or prophylaxis.
That's prophylactics.
It's prophylactic, right? But if you are in the arena of prophylaxis, regulatory hurdles are enormous, and the outcome measures are not yet clear. The big problem is when we talk about prophylaxis, we would need good biomarkers. Take cardiovascular disease as an example. In cardiovascular disease, right? Let's say we know that smoking is a risk to develop cardiovascular disease. In the moment when you have developed a myocardial infarction, you would not tell the patient stop smoking. You can still tell the patient stop smoking, but the therapy would be different. You have to now to target the myocardial infarction, have to treat that. But you should have treated earlier. But the question is, what can you then target? And in cardiovascular disease, there are biomarkers, for example, arterial hypertension, that is, I think there's agreement amongst all of us that arterial hypertension is a disease, but it's actually not a disease. It's not a disease, it's a biomarker, but it's so well established that it's being perceived as a disease, and FDA would agree and would say, if you now have if you now have arterial hypertension, it's a biomarker, and if you can intervene and can lower arterial hypertension, that's a good thing because it will have an effect on the end stage that you would like to avoid. We are not there yet. That's a big problem. That's one of the reasons why ProgStar was needed, because you have to show FDA that's the natural history, that's the end stage. We are earlier, we can follow, and there's a clear path, and we have to work along that path and would intervene earlier to allow to have a better outcome. But it's hard, and ProgStar was one attempt to make a difference in this respect.
As long as I'm alive, I will agitate you about this prophylactics because that's the way to go. But not to take more time, let's move to Corinne again.
Sorry, I'm back to a technical question. A quick question towards your gene tissue targeting vectors. So when you screen them, do you screen them through with the optics of doing subretinal injections or vitreal injections? And do you have different vectors for each type of injection?
No, it's a very good question. For the time being, since we are currently interested in diseases that affect the outer retina, we almost exclusively are working on subretinal delivery. We even work on new surgical methods of subretinal delivery, but it's all about subretinal. I think at IOB, of course, if there's an opportunity to go intravitreal, that's an easy thing. We would always welcome that. But for the time being, working on outer retinal disease, we believe that the time has not come, in my opinion, for outer retinal disease, not inner retinal, but outer retinal disease, to avoid subretinal. You have to do subretinal, in our opinion.
Okay, let's move to Kano.
Oh, hi. Wonderful talk. Yeah, if I am understanding correctly, you are also running the optogenetics clinical trial, right? Right. So can you comment?
The clinical trial, meaning that currently we're doing this iconic study retrospective that is preparing for the prospective observational study that should then lead immediately into the clinical trial.
Can you comment like what types of options you are using? Like there are various types of optogenetics agent, and also like as channel of sin or crimson, are they still need a very high intensity light and you have to use the goggles or something? Have you developed something that can be using ambient light, or what is that, how you stimulate?
Yeah, it's a very, very good point. I don't think that, and that is a more chemical cell biology question, but to my knowledge, there is very little wiggle room, and we will certainly not get anything beyond 3 log units dynamic range. So if that's the case, if you speak about typically 2 log unit and maybe 2.5 log unit dynamic range, if that's the case, you have to adapt, which means you either, if it's ambient light, sounds great, but that means that you then have to block light if it's relatively bright, or the other way around, you have to amplify. But I don't think that we will, for the time being, will or can avoid some sort of amplification, or maybe if ambient light would be sufficient for some of these general rhodopsins, that you need to at least modify and adapt to the specific situation. And that would mean that for all the approaches that I have seen, you need some sort of, let's call them goggles. This is what we are developing or having. But I doubt that that we will see such an approach that would happen without goggles or something similar.
I see. Well, I mean, for amplification, I think that there's a microbial option, like channel option or whatever option you're talking about. They don't have the amplification system unless you couple with G couple or something other protein types. So the amplification you're talking about.
Exactly. You have a set dynamic range, and now that's debatable. Our approach is that compensate for that lack of dynamic range by typically amplification. If you have something more sensitive, then it's the other direction, but typically amplification. And that would be accomplished by using a separate device that would project onto the retina.
I see. Okay, thank you.
We had a phenomenal presentation, so thank you for that. We had a fantastic discussion. So we now, Kathy, don't you worry, now ready for final comments. I would suggest maybe Professor Brown will just give it final thoughts.
Yeah, great. Thank you so much. Really enjoyed the talk. I mean, covering so many different approaches to dealing with these outer retinopathies, which I encounter in my clinic as well. I was wondering if you can comment on the deuterated vitamin A compound and its use in Stargardt's. and the progression of geographic atrophy. Was there any analysis that was done on central versus peripheral macular geographic area foci, areas that have fewer cones, for example?
Yeah, good point. The answer was that in the clinical trial, the inclusion criteria were agnostic of the acuity. These DDAF lesions simply needed to be within the imaged areas that you can easily capture by fundus autofluorescence. And that means typically within 15 degrees of eccentricity. So almost all of them were parafoveal or macular at least, yeah. Many of them were foveal, yeah.
That's great.
Sorry, I mean, you rightly specifically addressed the issue. Was there a data analysis of the directional spread of those lesions, either towards the fovea and away from the fovea? And it was, and there is a difference, yes.
Okay, yeah, very interesting. It'll be interesting to see those results whenever they do come out. And possibility of expanding to other geographic atrophy related conditions. Yes. So thank you so much for taking the time to talk to us today. I really enjoyed everything you shared.
Hendrik, I'll skip one student question. So let's go before Andrew. We kind of like introduce before Andrew, but it is done, so I don't know how to do it. Sam.
Hi, Dr. Scholl. A lot of my questions were already asked, but I was wondering if you comment on why some patients seem to have foveal sparing in their cones and why some RP patients do not.
I keep it brief. I do not know. It's an interesting thing. Why is there foveal sparing in some and why is there not foveal sparing in others? If we only knew, I mean, that would bring us to the question, why the macula in the first place, right? You look at ABCA4 expression and then you can ask why the macula, right? I mean, yeah, there may be some reasons, but I don't think they are sufficiently explain the phenomenon that the macula is affected and the periphery is much less affected. But it's a very good, it's an important observation we always make in clinic, and I have no good explanation.
Hendrik, don't you worry, he always asks tough questions. So thank you so much. What a delight.
Thank you very much. Thank you. Thank you. Enjoy the rest of the day. I'm already ready for the weekend here, so it's 6:30 p.m. Thank you.
Thank you. Thank you, everybody. Tomorrow is another day. That's what I say.